Hi everyone, I'm Christina Lingham from the Next Generation DX Summit. I'm thrilled to have the opportunity to speak with Giulia Siravegna from the University of Torino on her lab's unique approach to employing circulating tumor DNA as a non-invasive biopsy. She will be presenting an update at the Next Generation DX Summit Clinical Application of Cell-Free DNA Conference taking place August 19th through 20 in DC. Giulia, thank you for joining us.
Thank you.
Can you describe your approach for the recent study on the potential of circulating tumor DNA in evaluating response to chemotherapy in colorectal cancer patients?
Sure. Well, our study is based on previous evidences that tumor-derived DNA is shed into the bloodstream by cancer cells. And although the presence of circulating tumor DNA in blood of patients has been known for decades now, only recent advances in genomics have allowed the detection and quantification of cancer-related molecular alteration with high specificity and sensitivity. Our group is actually interested in precision medicine and in particular in colorectal cancer patients, and we initially realized that we could not understand the mechanism of acquired resistance to targeted therapies, like for example the EGFR blockade with anti-EGFR antibodies or BRAF inhibitors using only tissue biopsies for the inherent limitations. And that in that case, we will not be able to interrogate the whole heterogeneity of the disease. So we found that the tumor tissue, the genotyping of the tumor tissue will not comprehensively interrogate the heterogeneity of the disease. And so we moved into this non-invasive approach, the liquid biopsy. And we assessed whether the blood-based molecular profile could be used to identify actionable targets, to monitor drug resistance, and to track the tumor dynamic. And to do that, we use beaming, the digital PCR approach that we kind of build our own in our institute, and we lately use the next generation sequencing approach to obtain a more general profile of the circulating free DNA.
Can you explain how circulating tumor DNA is most likely to be used by itself and in conjunction with other tests in the standard of care for colorectal cancer?
Well, I think that the most likely application would be the monitoring of patients undergoing targeted therapies, the monitoring of the detection of the minimal residual disease with a more candidate approach, the diagnosis and the discovery of new actionable targets using NGS. Of course, the liquid biopsy approach should be coupled with the standard serum markers and with the imaging approach, but of course it will not completely replace the tissue biopsy, but for the reason I was explaining before. But of course I hope that it will be added side by side and easily repeated many times with the standard approaches.
What advice do you have for employing circulating tumor DNA in evaluating early therapeutic response?
Well, I think that the best way would be to collect serial blood draw during the treatment and also right before the starting of the treatment, because of the spike that we see in the very first weeks of treatment before the subministration of the cycle 1 of the chemotherapy. So this spike has been proven to be an early indicator of response, and for sure it will be helpful for clinicians to consider to switch to another therapy or to keep going with that therapy without the need of the CT scan evaluation.
What are some of the most exciting applications emerging for liquid biopsies?
Well, I think that the most exciting ones are this heterogeneity assessment, the heterogeneity discovery of and the complexity of the clones dynamics in the tumor, and together again with the assessment of the minimal residual disease after surgery, and so to decide whether the patient would need additional therapy or if we can spare them to additional toxicity and so on. We for sure will apply the liquid biopsy approach systematically in the frame of clinical trials in colorectal cancer patients first, but also in other settings like lung cancer, melanomas, pancreatic cancers, and this will be helpful to monitor together with the clinician how the clinical trial is going, and to have a more comprehensive view of the treatment benefit, and in order to be able to monitor virtually continuously the molecular evolution of the disease through the liquid biopsy.
Giulia, tell us about the results from the recent liquid biopsy study published in Nature this week.
So we performed this very exciting study. We found that circulating DNA could be used throughout the clinical management of the colorectal cancer patients, and we in this way find a way to gather in real time updates on the molecular landscape of the disease. So first we determined the molecular profiles of this patient using blood, and we assessed like a concordance study between the genotype in the blood and the mesh tissue in over 100 patients. And then we applied the next generation sequencing technology to perform the analysis on the resistant patient, resistant to anti-EGFR therapy. And we found many alterations that were responsible for the acquisition of resistance, let's say NRAS mutation, KRAS mutation, HER2 amplification, MET amplification, and so on. Also some novel EGFR extracellular domain mutation. And we then followed patients after the acquisition of resistance, and we find that the KRAS clones actually declined in the circulation when the selective pressure of the anti-EGFR antibody was withdrawn. So this was very interesting also because those KRAS mutant patients do not have any further lines of treatment when they acquired this mutation at resistance. So it was very interesting to observe how this KRAS allele dropped down, and so how this patient could be rechallenged with the same anti-EGFR therapy, achieving again a clinical benefit.
Lastly, what is unique about your approach? How did you become a leader in this field?
Well, we started the project in 2010, and I think that the best thing was that we've always believed in this approach, and because we believe that this approach would be very useful for patients, also to study the complexity of the colorectal cancer, but moreover for the patient. And although we knew that working with circulating free DNA is very challenging because it's very poor quality, it is not high as concentration in the circulation, but from a technical point of view, was very challenging. But with the 5 years experience now, we've built our own facilities with our own technologies, and we set up, as I was saying, the beaming technology, and we exploited other digital-based PCR approach and the NGS with our own targeted panel of 226 genes, which is able to capture and sequence the whole coding region of more than 200 genes. And so with our own efforts, we were able to analyze with a very high sensitivity and specificity the circulating DNA. And of course, all of these would not be possible without the collaborators in a lot of hospitals in Italy and all over the world, and in particular with our collaborators in Guarda Hospital in Milan, who provided patients and collected very carefully all the samples.
Giulia, thank you for your time and insights today. That was Giulia Siravegna of the University of Torino. She'll be speaking at the Clinical Application of Cell-Free DNA at the Next Generation DX Summit taking place on August 19th through 20 in DC. If you'd like to hear her in person, go to nextgenerationdx.com for registration information and enter the key code Skype. I'm Christina Lingham. Thank you for listening.